everyone. In today's lecture, we will learn about the topic entitled Microbiological Approach to a Plastic-Free World. Now, before going into details, we will go through the objective of this lecture. First, to study about plastic waste and its effect on the environment, including on the living organisms. Secondly, to study about the available techniques of plastic biodegradation. And thirdly, to study about the role of microbes in plastic biodegradation to prevent microplastic accumulation. Plastic usage has transformed our life in various ways. The production and utilization of plastics are always increasing due to their rising demand. They are inexpensive, strong, lightweight, corrosion-resistant, hard duration, and electrical insulating properties and have high thermal properties. 500 billion to 1 trillion per annum polythene covers have been under regular use worldwide. The useful breakdown of plastic bags takes more than 1,000 years. Plastic causes global warming and pollution, not only as a major issue of waste disposal, but it also releases a high quantity of carbon dioxides. Now we come to microplastic formation and its impact on animals and birds. Microplastic is being formed due to the photodegradation of large plastic by the sunlight, converting it to be even more toxic, thereby contaminating the sand and the water. It can be accidentally be consumed by the terrestrial and aquatic animals and enter the food chain. Especially microplastic has become a mandatory product in the food chain of marine biota nowadays. In the aquatic ecosystem, the waste is the principal risk factor for marine animals. It causes abdominal obstruction in the fishes, birds, and other aquatic and terrestrial animals. As per the report, plastic contaminants dumped in the aquatic environment affect at least 276 species, including all animals, especially 44% of the seabirds and 86% of the sea turtles. The postpartum of the dead terrestrial animals have been reported to have polyethylene carry bags in the intestine, which is considered to be a major root cause for its death. These polythene has blocked the entire alimentary canal, and its secondary toxin and intermediate digestive products have led to the toxicity which led to the death of these animals. It has been reported that the polythene obstructed digestive functions such as fermentation via blocking the other components from blending with the digestive enzymes and juices, resulting in indigestion. The blockage of the opening between the reticulum and the omasum due to the obstruction of polythene becomes highly fatal for the animal if the polythene is not being removed from the ecosystem. The digestive salts react with the polythene and form complexes which obstruct the digestive tract's food passage, leading to irritation in the rumen and pain due to immunosuppression. The plastic accumulated in the alimentary canal reduces the immunity and leads to the secondary infection such as hemorrhagic septicemia in the dead cow. At a global scale, nearly 10% of the municipal waste contains used plastic covers and materials. Now, based on a survey, every year 100,000 tons of plastic products have been dumped into the ocean, sea, and aquatic environment, resulting in various animal death. During the biodegradation of plastic, it enters into the food chain and produces secondary toxic material and toxins, which are fatal for the aquatic animals. Now, natural decomposition is a common phenomenon in which complex organic matter is transformed into a simple organic substance by living organisms, specifically by the microbes. The microorganism obtains nutrients from the plastic via enzymatic degradation process. Plastic waste serves as the source of energy and carbon required for their growth and development. It is a key part of recycling materials by the natural ecosystem. The plastic materials can be degraded either under anaerobic or aerobic conditions. Aerobic bacteria utilizes oxygen as an electron acceptor and breaks down the complex organic chemicals into simpler forms, often producing carbon dioxide and water as the end product. Aerobic and anaerobic decomposition acts as a major environmental component for the natural remediation of contaminants at many harmful waste sites. In anaerobic decomposition, the microbial mechanism degrades the organic pollutants without the involvement of oxygen. Certain anaerobic bacteria involve carbon dioxide, manganese, sulfate, iron, nitrate, organic chemicals during the degradation process as an electron acceptor to form simple products. Algae are known to colonize on artificial substrata like polythene surfaces in sewage water, and these colonizing algae were found to be less hazardous and non-toxic. Adhesion of algae on the surface will initiate the biodegradation, and their production of lignolytic and exopolysaccharide enzymes is the key for plastic biodegradation. The algal enzymes present in the liquid media interact with macromolecules present at the plastic surface and triggers the biodegradation. The polymer is utilized by algae as a carbon source, since the species growing on the polythene surface were found to have a higher cellular contents like protein and carbohydrates and a higher specific growth rate. Additionally, surface degradation or breakdown have been seen clearly on the transverse section of the algal colonized PE sheets. 5 methods of biodegradation includes fouling, corrosion, hydrolysis and penetration, degradation of the leaching components, as well as pigment coloration via diffusion into the polymers were observed in the past works. Polymer degradation is defined as any alteration in its physical or chemical properties due to the environmental factors, including light, heat, and moisture or biological activity. There are 3 types of polymer degradation methods: photodegradation, thermo-oxidative degradation, and biodegradation based upon the factors involved. Now, it was reported that microorganisms such as bacteria, fungi, and algae could degrade polymer materials through their metabolic activity. The so-called biodegradation without the involvement of heat energy under aerobic or anaerobic conditions. In the aerobic biodegradation, the end products produced were carbon dioxide and water in the soil composite method. The anaerobic biodegradation of landfills and sediments includes methane, water, and carbon dioxide as the end products. Now, usually, it is a complex process to produce water and carbon dioxide from the long chain polymer, which needs various steps and different microbial activity. In each step, a particular microbial community will break the polymer into granules, and the others will utilize the monomers and excrete them. The microbial community of the detritus food chain utilizes the excreted waste. It is an eco-friendly, cost-effective, globally accepted method. However, the efficiency is moderate. A study in the biodegradation of 3 types of polythene carry bags have been experimentally tested in aquatic ecosystem. Ordinary polythene along with 2 other types of plastic carry bags were subjected for degradation and found that 2% of the degradation at the surface of the biodegradable polythene carry bags have been reported after 40 weeks under experimental conditions. The degradation of standard polythene was reported to be negligible. Now, with this, we come to our first quiz. The question is, which of the following is not a method of biodegradation? The options are number 1, fouling, number 2, corrosion, number 3, hydrolysis, number 4, osmosis. And the answer is osmosis. Now, the second quiz question is, microorganisms such as bacteria, fungi, and algae could degrade polymer materials through their metabolic activity, which is known as number 1, bioformulation, number 2, biodegradation, number 3, assimilation, and number 4, fermentation. And the correct option is biodegradation. Cut. Now we come to our next topic, environmental and economic impact. The Ecosystem Service has reported the substantial negative impact of plastic waste on experimental recreation, where the aquatic ecosystem, including the coastline, has been exposed to plastic. This directly contributes to the economic costs impacted by these plastic wastes, especially in the cleaning up process for the ecosystem. The presence of plastic waste on the shore may be one of the main key reasons why visitors will spend less time in these environments or avoid certain sites, which will eventually lead to the decrease of tourism revenue in those tourist spots. Other direct consequences caused by the presence of plastic wastes are a range of unexpected injuries. Example, sharp debris, entangled in nets, and exposure to unsanitary items. Detrimental to physical and mental well-being. Example, social interaction, family bonds, and physical activity. And intensively colonized by a wide range of opportunistic species. Now, plastic degradation by microbes. How they are degraded and in what way we can analyze the extent of degradation by microbes. There were various reports available on polythene degradation by microbes. The bacterial isolates from the dumping areas were used and tested for the characterization of tensile strength, surface corrosion, and percentage of weight parameter analysis. The sample exposed under the experimental condition for 3 months with regular shivering of the polythene disc showed surface corrosion, reduction in tensile strength, and a maximum rate of 12.5% of weight loss by Pseudomonas species and Bacillus cereus species. These were the 2 strains that were identified using biochemical tests and morphological keys actively involved in this degradation experiment. Biodegradation of degradable plastic polyethylene by the fungi phanerochaete and the bacteria Streptomyces species have been reported. The methods used for testing plastic degradation are mainly molecular weight distribution, weight loss, change in tensile strength, and change in the percentage of elongation. The type of microorganism used was mainly lignocellulose degrading fungal and bacterial strains. The biodegradation potential of microbial isolates obtained from various soil sources such as agricultural, sludge zone, energy park, sewage water, medicinal garden, etc. were evaluated. The experimental sample used for analysis were plastic cups and polythene bags, which were incubated with fungal and bacterial strains for almost 31 days. The maximum biodegradation rate shown was 12.25% and 12.5% by the fungal strain Aspergillus niger and the bacterial strain Streptococcus lactis respectively. Other microbes such as Pseudomonas, Aspergillus nidulans, Bacillus subtilis, Proteus vulgaris, Staphylococcus aureus, Aspergillus niger, Streptococcus lactis, Aspergillus glaucus, Aspergillus flavus, Penicillium, Micrococcus luteus also showed the potency for degrading the plastic cups and polythene bag. Biochemical and morphological tests were used for the identification of microbial strains. The rate of biodegradation has been evaluated using the loss of the weight method. Now, it was investigated that the biodegradation potency of microbial isolates from the Baltic Sea and tested with various polymers. Samples include clear polyethylene with 5% of starch, modified polyethylene films containing 8% and polyethylene with 20% pro-degradant additives. Tensile strength, change in weight, and morphology of polymer were the methods used for evaluating the rate of biodegradation. The samples were incubated in the Baltic seawater for 20 months. The polyethylene blends showed only minimal degradation during the summer and the winter season. Now, a study to bioremediate the plastic waste in municipal solid waste were done by isolating the microbe from the soil samples collected from municipal solid waste compost, from which 250 strains were isolated and screened with probable identification for plastic degradation, and found that the fungi Trichoderma viridi showed active bioremediation potential. The fungal strain was incubated for a period of 60 days and analyzed for average weight loss, and the strain was identified by using biochemical and morphological tests. The production of cellulose enzyme and the loss of weight were the parameters used for calculating the bioremediation rate, and found to have a weight loss of 20.10% and 33.35% with plastic plates, and 33.35% as equal as the plastic plate with compost pile. Our next topic is microbial mechanism of polythene degradation. Decomposition of polythene has been initiated with its attachment to the microbial cell surface. Bacteria, including various Streptomyces species and fungi, secrete extracellular enzymes that can facilitate the decomposition of polythene. In fungi, the lignolytic system's extracellular enzymes contain lactases, oxidase enzymes, and catalase that produces the extracellular hydrogen peroxide. Based on the microbe, culture conditions, strains, and enzymes secreted, the degradation of plastic may vary in nature. The decomposition of lignin involve three main enzymes: the manganese peroxidase, lignin peroxidase, and phenol oxidase, which includes copper as a cofactor and is known as S-lactase. Apart from that, microalgae can be genetically modified to a microbial cell factory, which is capable of producing and secreting plastic degrading enzymes. For example, green microalgae Chlamydomonas reinhardtii was transformed to express PETase, and the cell lysate of the transformant was co-incubated with PET, resulting in dents and holes on the PET film surface as well as on TPA, which is the fully degraded form of the PET. P. trichonertum was successfully used as a chassis to produce PETase, which showed catalytic activity against PET, and the copolymer polyethylene terephthalate glycol. These studies have provided a promising, environmentally friendly solution to the biologically degradable PET using microalgae via synthetic biology. Now, in nature, there is also a list of secondary products which are formed from the polyethylene from polythene degradation. Now, based on this lignolytic enzyme's potency, they have been used widely in different industries such as chemical, textile, fuel, agricultural, food, cosmetic, paper, and are used to remediate xenobiotic compound dyes. During the degradation of phenolic compounds, the lignin materials are degraded via oxidation under MnO2 and H2O2. Manganese dioxide oxidizes monomeric phenols and converts Mn2 to Mn3, and Mn3 further oxidizes the synthetic lignin and phenolic lignin dimers via the formation of phenoxy radicals. The production of various secondary products from polythene degradation is based on biodegradation condition. Under aerobic conditions, carbon dioxide and water is the end product produced, and anaerobic degradation leads to methane in case of methanogens, water and carbon dioxide as the final product. However, hydrogen sulfide gas is produced under the existence of sulfate reducing bacteria. Now next we come to the toxicity of biodegradable plastic. Pure plastics are less toxic due to their relative chemical inertness and insolubility in water. Adipates and phthalates are plasticizers which are used as an additive in several plastic products such as polyvinyl chloride or PVC to change its tensile strength and percolate these compounds out of the product in trace amount. Polystyrene in the food containers has been predicted to be leached and enters into the human body, resulting in a hormonal imbalance, highly carcinogenic, and other adverse effects in the living organisms. The entirely produced parent polymer are free from toxicity, but the monomers used during the production processes are highly toxic. The effect of biodegraded polythene was monitored on seed germination in plants, including soybean, safflower, groundnut, sesame, and sunflower. During the observation, the pre-treated seeds show a decreased percentage in seed germination. The degradation of polythene bags, cups were studied using Pseudomonas aeruginosa, Streptomyces species, Aspergillus niger, Staphylococcus aureus, Rhizopus species, and the biodegraded polythene toxicity level. Carbon dioxide gas is the main product produced during the degradation of polythene. The granules from the bio-treated polythene showed adverse effect on the roots, which leads to abnormalities in the production of polysaccharides, proteins, and also nutrient uptake. Protein was the only product produced in Nocardia asteroids bacterium. The production of carboxylic acids, ketones, and aldehydes in the smoke of film extrusion of low-density polythene, LDPE, was observed. Now, what we have seen is that awareness must be created about plastic pollution and its adverse effects on living organisms. Mass level, through monthly campaigns and eminence of polythene pollution, should be updated area-wise to create an awareness among the public and the common people. People should be encouraged to use eco-friendly products. Methods for proper disposal of plastic must disseminate among people using all available media platforms, which includes the social media. Selection of appropriate microbial strains, adapting suitable in situ and ex situ remediation techniques, continuous monitoring of remediation site, and proper maintenance, such as providing proper aeration, nutrients necessary for microbial growth, and physico-chemical conditions are highly required. Genetic, molecular analysis for identifying gene responsible for producing plastic degrading enzymes and recombinant DNA technology can improve and accelerate remediation of plastic waste and its disposal. The awareness should be highly created at the school level by guiding the students to properly separate the biodegradable and non-biodegradable plastic waste before its disposal. Microalgae contributes to the biodegradation of plastic waste with its enzymes that weaken the chemical bonds of plastic polymers. The attempt of using microalgae to convert these plastics into metabolites such as carbon dioxide, water, and new biomass is of great interest. However, this field is relatively new and more study is required. Considering the variety of plastics investigated, more biodegradation studies employing algae must be carried out, and the efficiency must be evaluated. Moreover, it has yet to be researched on how microalgae can specifically degrade microplastics. Now, with this we come to the end of this lecture. What we have seen is that microbes can be utilized for polythene degradation in various ways. Various microbes have secreted various kind of enzymes for degrading these plastics. However, when we are using microbes for degrading polythenes, we must keep one thing in mind, that we cannot use any kind of harmful, toxic or pathogenic microbe for an environmental aspect. Thank you.